Please join me in welcoming Professor Noam Stern-Ginossar. She comes here from the Weizmann Institute, so currently she's also at the CZ Biohub. She did her Ph.D. at the Hebrew University and then did a postdoc with Jonathan Weissman at UCSF. And since then, she has gone on to use deep sequencing techniques, ribosome profiling, which I think have been extremely exciting techniques like all across science. And she's using them to look at CMV, Cytomegalovirus, and to look at how latency and infection can be predicted, analyzed, measured, all of these things using this really exciting technique of deep sequencing and RNA sequencing. So yes, please all help me in welcoming Noam.
Thank you. Don't see my presentation. What's that?
Oh, the guy.
Is it this?
Yeah.
No, I share screen and I showed it before. No.
I think it is. Does anyone know how to use this thing?
It was fun like 2 minutes ago. Just a second.
I think
I was somehow disconnected from the room.
Hey, are you in the Zoom?
I was, but now I'm not. So I was connected, I swear. Okay.
Do you want to tell her the Zoom ID, meeting ID?
She's connecting.
Okay, yeah, she's there.
And then can you share?
Sure.
Because there's buttons here.
Okay, yes. Sorry for that. Okay, so thanks a lot for the introduction and for the opportunity to present some of the stuff we are doing. So as Jimena just said, I'm coming from the Weizmann Institute, but actually I'm spending the next year from the summer at San Francisco Biohub. So if anyone wants to interact, I'm actually quite close here, and it's a really great place and it's really fun to be there. Okay, so I gave the title a widely encoded high-resolution screen of Cytomegalovirus host dependencies, and this will be the second half of my talk. And it's not public, but I wanted to give just an overview on the virus we are really interested in, and this is CMV and herpesvirus. Fascinating. They are found everywhere, likely infect all vertebrates and even some invertebrates. These are very species-specific, so human herpesviruses infect only human, they are endemic to human. These are large double-stranded DNA viruses with huge genome that replicate in the nucleus. And one of their defining features is that they persist for the lifetime of the host. And I think this is a really an amazing property. And this property was designed along many evolutionary years. So these are really, really well adapted to the host. And I think this makes them an amazing tool to study viral host interaction because nothing is done by mistake. So specifically, CMV is the largest known human virus, almost the half size of bacteria, 240 kb the genome. It's endemic to human, and like all other herpesviruses, it persists to life. So once you get infected, you never get rid of this virus. In addition, it's an important pathogen. It's in fact the majority of the human population, like most herpesviruses. And although it's asymptomatic usually asymptomatic in every individual, it's a major cause for birth defect because it's one of the only viruses that can cause the placenta during pregnancy and infect the fetus. And actually, we don't really understand how it's doing it. And second, it can cause severe disease in immunocompromised individuals. So it's a really fascinating pathogen. And so what my lab has been interesting in the last almost 10 years is first to, and I think this is what Jimena was relating to try and understand better how CMV and herpesviruses commandeer the gene expression machinery in the cell, specifically the ribosome. But I decided actually to share this today, actually a story about SARS-CoV-2. So we used all these tools that we built to characterize interference with herpesviruses and to understand how SARS-CoV-2 is doing it. So this is the most recent work, so I'll talk about that. And then we use unbiased gene expression methods, and specifically single-cell RNA-seq, to try and understand better the molecular properties of CMV latency. Sadly, I'm not going to talk about it now, but if anyone is interested in chatting about it, I found CMV latency as one of the most fascinating fields. I think there's still a lot to be discovered. And lastly, we used ribosome profiling-based approaches to better characterize the genomic structure of herpesviruses and revealing really high complexity, much more than what we anticipated before. But the big challenge for many years was, okay, so we have all these coding sequences, we also apply this method for SARS-CoV-2, but what are these new open reading frames are actually doing in infection? And are they doing something? And for many years we were struggling with this, and in the second part of my talk I'm going to describe a new method we developed, which I think is going to help us to address this question in a systematic manner. Okay, so I'll start by shortly introducing the life cycle of SARS-CoV-2, as I assume most of you know it. So once the virus enters the cell, it injects very long single-stranded positive RNA genome into the cytosol. And then what this genome needs to do is to recruit cellular ribosome that will translate the first very long open reading frame, which actually generate two long polyproteins that are then politically cleaved. They are politically cleaved to generate the 16 non-structural proteins, many of which will generate the RNA-dependent RNA polymerase that will either replicate the genome, generating more copies of the genomic RNA, or generate a series of subgenomic RNAs that share the same 5' sequence. And these subgenomic RNAs again need to recruit cellular ribosome to make the accessory and structural proteins that will allow the virus to finish its life cycle. And the step my lab is super interested in is this critical step in which the virus needs to recruit cellular ribosome to translate viral mRNA. And this is critical in all viruses, DNA or RNA. Okay, so when the pandemic started, we asked ourselves if and how SARS-CoV-2 interfere with host protein synthesis. And specifically, does it cause host shutoff? So host shutoff is a common phenomenon that is applied by most viruses, and it's thought to give the virus two potential advantages. First, if you shut off host protein synthesis, you theoretically allow a better translation of your own viral transcript because there is competition between viral RNA and cellular RNA. And second, if you block the cell ability to generate new protein, you block its ability to mount an efficient antiviral response. And many viruses develop diverse mechanisms to achieve it. So we have viruses that interfere with transcription, different processing steps in the nucleus, viruses that block export out of the nucleus, viruses that directly manipulate the translation machinery, and viruses that degrade cellular RNA either in the cytosol or in the nucleus. So we asked ourselves, what is SARS-CoV-2 is doing? And initially, this was the beginning of the pandemic. We calibrated conditions in which we can efficiently infect caliph-free cells. These are lung adenocarcinoma cells. And then we used a poromycin homolog, which is called OPP, which basically incorporates into immunity-generated nascent chains. And then by click chemistry, we can label these nascent chains. So this is very similar to S35 labeling, but it's just not radioactive. And then we can quantify by FACS, basically the amount of translation these cells are having. So you can see here very clearly in uninfected cells, we have nice amount of translation. And as infection progresses, 8 hours post-infection, there is more than 4-fold reduction in overall translation. So this shows SARS-CoV-2 is massively interfering with translation. But these are global measurements. We can't say what happens to viral RNA or cellular RNA. So to try and address the division between these transcripts, we apply ribosome profiling. So again, we take caliph-free cells, infect them with SARS-CoV-2, and then collect samples for ribosome profiling, basically capturing ribosome-protected fragments, which give us an overall snapshot of all the translation events, both viral and cellular. And in parallel from the same dish, we also do RNA-seq, so we have the measurements of RNA expression, and we done it in uninfected cells in 3, 5, and 8 hours post SARS-CoV-2 infection, everything in replicates. Okay, so first we looked what happens to the translation of viral and cellular gene, and we calculate something that we call translation efficiency. So we take the number of ribosomes we isolate, which reflect how well this transcript is translated, and divide it by the number of the mRNA molecules. So this is like a normalized translation efficiency. And this is the graph we see. So in the black, you basically see all human genes. So it looks like a line. And in the colorful dots, you can see the main viral transcript. And the surprise is that we actually didn't see any major peripheral translation of viral RNA. They looked like cellular transcripts. So this made us focus more on the mRNA itself. And so if we do RNA-seq and total RNA-seq without any polylase selection, and we sequence the samples, this is what we get. You can see that within 8 hours, there is massive production of viral RNA, and more than 90% of the mRNA in the cell is coming from the virus. But what we also noticed is, if you see in blue, we actually see reduction in the amount of cellular transcripts. So it's very hard to understand from global measurements of RNA whether this reflects true changes in. So it's hard from steady-state measurements to get something about the half-life of transcripts. But we knew that if viruses interfere, so if SARS-CoV-2 interfere with a cellular mRNA production, it usually occurs in a certain compartment. So what we've done, we took all the cellular transcripts and binned them according to the subcellular localization. So these are the most nuclear transcripts, and these are the cytosolic transcripts. And we look how each of these bins is changing in SARS-CoV-2 infection. And I hope you can see that the transcripts that are changing in infection are always the cytosolic transcripts, whereas the nuclear transcripts are not drastically changing. So this made us hypothesize that SARS-CoV-2 actually degrades cellular RNA in the cytosol. And so to directly address that, we used a method, very powerful method that is called SLAMseq. So I'll walk you through the experiment. So we take, again, calorie-free cells, we infect them or not infect them with SARS-CoV-2, and then 3 hours post-infection, we use a uridine homologue, which is called 4SU, which will now incorporate into newly synthesized RNA. And then we harvest the RNA at 1, 2, 3, and 4 hours post-labeling. We then extract the RNA and use iodoacetamide to alkylate the 4SU. It's something that by sequencing will look like cytosine. And then by RNA-seq, we can identify for every read whether it's newly generated RNA or old RNA based on the U to C conversion. And from this U, we can calculate basically the half-life of all cellular and viral transcripts. And this experiment actually gave a very clear answer. So you can see here the half-life in infected cells of all cellular RNA versus the half-life in uninfected cells. And there was a major reduction in the half-life of almost all transcripts. And we can show also on a genome-wide level that this reduction in RNA stability occurs mostly for cytosolic transcripts, whereas nuclear transcripts are much less sensitive to reduction in their half-life. So the conclusion was that SARS-CoV-2 can induce massive mRNA degradation of the host. And the next big question was, who is responsible for this degradation? And there was one immediate suspect. We didn't have to work really hard. And this protein is named NSP1. So NSP1 is the main shadow factor of all alpha and beta coronaviruses. And within 6 months after the pandemic started, there were already papers, structural papers, that shows that NSP1 physically blocks the entry channel into the ribosome, basically preventing mRNA from being translated. There was actually old literature from the lab of Shinji Makino in UTMB and the original SARS that suggested that NSP1 can also interfere with mRNA half-life. So we wanted to see if NSP1 indeed has the capacity to degrade cellular mRNA, and we used a very simple reporter assay. So we cloned GFP with a control 5' UTR, and then expressed it either with mCherry as a control, so of course the GFP is expressed, or together with NSP1, which really blocks the GFP expression. This was shown by others because it blocks translation. But what we could see that this is also associated with major reduction in the mRNA level of the GFP, basically proving that NSP1 can degrade cellular RNA. And then amazingly, if we take the 5' leader sequence of SARS-CoV-2, which is shared by all viral transcripts, and we fuse it to the GFP as a 5' sequence, then we can rescue the GFP expression and also rescue the RNA from degradation. So basically, we think NSP1 degrades cellular RNA, and viral RNA escape this degradation through their unique 5' UTR. And we think this can explain how within 8 hours post-infection, 90% of the mRNA in the cell is actually coming from the virus. So these were all very global measurements, right? Looking at globally what happens to mRNA, but the ribosome profiling and RNA-seq actually give us also individual measurements on the transcript. So next we was wondering whether there are genes that show specific changes in the relative translation efficiency along SARS-CoV-2 infection. So for every gene, we calculated its translation efficiency, so footprint divided by the mRNA, And we clustered the genes that shows most drastic change in the translation efficiency along SARS-CoV-2 infection. And this actually revealed a really interesting signature, is shown here. So the genes that shows the most drastic reduction in the relative translation efficiency were actually genes that the mRNA was induced. So these are actually the only innate immune genes. You can see the mRNA goes up, but the footprints are not changing. So if the mRNA goes up and the footprints are not changing, we see relative reduction in the translation efficiency. But what this actually suggests is that for some reason, newly generated transcripts never make it to the ribosome. Right? So they are generated, but they never engage with the ribosome, so we don't get footprints. And one possible hypothesis for why is this happening is maybe SARS-CoV-2 is actually blocking nuclear mRNA export. So if mRNA doesn't go out of the nucleus, it will never engage with the ribosome. So to test that, we again infected cells or not infected them with SARS-CoV-2, and then did a subcellular fractionation, actually using a protocol that Noam was calibrating in his Ph.D. And using regression, we can calculate for every transcript, it's a cyto and nucleus ratio, and this is what's plotted here. So basically, you can see that indeed, in infection, we have major retention of cellular mRNA in the nucleus, and specifically innate immune transcripts here in purple are heavily enriched in the nucleus, and we think this basically explains why they never make it to the ribosome. So to conclude this part, we basically showed that SARS-CoV-2 really efficiently block host protein synthesis using 3 potentially independent approaches. So we know it blocks the ribosome, NSP1 blocks the ribosome. We show NSP1 can degrade cellular RNA, and we could show that likely SARS-CoV-2 inhibits nuclear mRNA export. So this was really satisfying and interesting, and we got a nice publication, but there was actually several questions that raised from this. The first one was, do these really represent distinct functions? So I showed you that people showed NSP1 blocks the ribosome. We show it accelerates mRNA degradation, and actually concurrently with our publication indicating there is blocked nuclear mRNA export, it was shown that NSP1 can block nuclear mRNA export. And we know these gene expression pathways are connected. So if you block translation, it will affect stability. If you block export, it has effect on stability. So we're wondering, are these really independent function of NSP1, or maybe it has one main function and everything is just happening downstream of this function? So this was the first thing. And the second, what is actually the importance of NSP1 during SARS-CoV-2 infection? It's multi-functional protein, what it's for. Okay, so I'll use the next few minutes to answer this. So to try and understand if these are independent functions, we generated 2 mutants of NSP1. The first one we called NSP1 delta RB. Basically, it's missing 10 amino acids that prevent its binding to the ribosome. I'm not going to show you the data, but you'll have to believe me. And the second one is 2-point mutation, or maybe I should say NSP1 is a very short protein, 180 amino acids. It has a big end globular domain and a short C-terminal domain that actually blocks the ribosome. So we had this 10 amino acids in the C-terminal domain. This prevents ribosome binding. And then we had 2-point mutation in the N-terminal domain that in the original works from Shinji Makino, he suggested that these amino acids are important for the RNA degradation. So we call this one NSP1-CD for cleavage defector and NSP1 delta R. Okay, so now we take these mutants as well as the wild type and control and transfect it to cells either with the control GFP or with the GFP that has the 5' mutation from the virus. I'm going to show you just the results from the control GFP for simplicity. And so you can see that, as I showed you, so this is the control. We see GFP expression, now it's by fax. If we transfect with the wild type NSP1, there is major reduction in the GFP. If we have the delta RB, we don't have much interference with the production of GFP. And then with the NSP1-CD, we have a mediocre phenotype, so some reduction in translation. My Zoom is hiding me there. Okay, you see the mRNA here, right? I just don't see it. But basically, you can see that the wild type virus reduces the mRNA, but the delta RB doesn't affect the RNA at all. So we know the RNA degradation is dependent on binding to the ribosome. And the delta CD, although it still blocks translation, it doesn't affect the mRNA. So our conclusion from this is that RNA degradation requires ribosome binding, but is likely independent because we can have a mutant that still binds the ribosome, but doesn't cause RNA degradation. And then we wanted to see the effects on degradation, RNA degradation, and nuclear export. I'm not going to show you the results, but basically what we've done, we took these constructs, transfected them into cells, and did SLAM-seq, and we could show that only NSP1, the wild-type one, caused RNA degradation, so this really fits the reporter results. And then we repeated the subcellular fractionation, and we could show that actually all the wild-type and the two mutants, all of them can block nuclear mRNA export. And this was really important result because it was really hard to understand if there is a nuclear mRNA export to block, or is this just a consequence of the degradation that will reduce the amount of cytosolic RNA. And this experiment actually proved that NSP1 indeed has ability to block mRNA export, and it seems to be independent of its ribosome activity and degradation activity. Okay, so next we wanted to see what NSP1 is actually doing in infection, and we teamed up with the group of Shinji Makino that made for us a SARS-CoV-2 virus that has a deletion in these 10 amino acids, which prevent it from binding to the ribosome. So we took this mutant-infected CALO3 cells, and first wanted to see what happens to host shuttle. So we used again the OPP experiment to label overall translation. And as I showed you before, 7 hours post-infection, there is major reduction in overall translation when we infect with the wild-type virus. But if we infect with the mutant viruses, in this virus just have a 10 amino acid deletion in NSP1, you can actually completely rescue the shut-off effect. So there is no interference with the overall level of translation. And then if we do the SLAM-seq and measure RNA stability, you can see again that compared to the mock, the wild-type virus, we have major reduction in cellular mRNA half-life, but with the mutant, there is no effect on cellular mRNA half-life. So this shows that NSP1 is really the main protein that is responsible for RNA degradation in infection. So basically now we have a virus that doesn't shut off host protein synthesis. So next was, okay, what happens to the virus? What happens to viral protein production? And we're actually really surprised to see that nothing happens to viral protein production. So if you infect in IMOI and look, for example, at the SARS-CoV-2 nucleocapsid protein, we don't see major difference between the wild type and the mutant virus. And then we did growth assays, of course, but we did it in 2 different cell types, and this will be important. We did it both in viral cells and CALO3 cells. So viral are green monkey kidney cells that lack interferon. This is why everyone likes to work them. And then CALO3 cells are the lung adenocarcinoma cells. So let's start with the viral cells in blue. When we go the wild type and the mutant virus, we don't see actually any major changes in the viral progression between the mutant and the wild type. And we do RNA-seq in the parallel time points. You can see that there is also a nice accumulation of equivalent level of viral RNA. But if we do the same experiment in CALO3 cells, you can see that the mutant virus is lagging behind the wild-type virus, and you can see it also in the RNA-seq. So the big difference between CALO3 cells and viral cells, except for the fact these are monkey cells and these are human cells, is that viral cells are lacking the interferon response. And so just few words about the interferon response. So when a virus infects a cell, we have a series of pattern recognition receptors that can identify the infection, and then they lead to the secretion of type 1 interferon that will act both autocrine and paracrine manner by upregulating by the JAK-STAT signaling and upregulating a series of interferon-stimulated genes that give the cells, the infected cells and neighboring cells, this antiviral state. And indeed, if we look at the RNA-seq data, we can see that the major difference between cells infected with the wild-type virus and cells infected with the mutant virus, in the case of CALO3 cells, was the major reduction of interferon-stimulated genes, specifically in cells infected with the mutant virus. So to test if this is really what explains the difference between mutant and wild-type virus, we repeated the growth assays in CALO3 cells, but now we use the inhibitor of the JAK-STAT signaling, which is called ruxolitinib. And so what you can see, again, in red first, when we don't treat the cells, again, we can see that the mutant is lagging behind the wild-type virus. But if we treat these cells with ruxolitinib, we can see that we will rescue the mutant virus. So what this really shows and proves, and was quite surprising to me, is that actually the most critical role of host shutoff, at least in SARS-CoV-2 infection, is not to support viral protein production, but actually to inhibit the cellular immune response. So to conclude this part, we basically show that the NSP1 is a multifunctional protein. It blocks translation in degrade RNA and block mRNA out of the nucleus, but it really has one major target, and this is interferon beta, type 1 interferon. So when wild type virus is infecting cells, there is hardly any production of interferon, and then when viruses will bud out, they will meet naive cells and they can efficiently infect them and propagate. But if we have the mutant virus that doesn't degrade RNA and doesn't block translation, then these initially infected cells can produce interferon, and then when the virus will bud out, it will meet cells that were actually upregulating the ISGs, and this basically blocks the virus propagation. And I didn't have time to show it, but we also use these in collaboration. We use these viruses in hamster model, and we could show like other people that if you use a wild type virus, you get sick hamsters. But if you use the mutant virus, the hamsters are actually completely healthy, and there is major reduction of interferon-stimulated genes very early on in infection. It's really also important for SARS-CoV-2 pathogenesis. Okay, so I'm now completely going to switch gear and moving back to my favorite virus, CMV, and basically trying to explain to you or tell you or share with you a new approach we developed to try and answer something that we were struggling for a long time. And so I told you we used ribosome profiling to really decipher the complexity of herpesviruses. And we annotated also for SARS-CoV-2, by the way. And we annotated many novel open viniframe. But the big challenge was, how can we better define the role of these genes? And more globally, how can we define the role of genes, cellular genes or viral genes during infection? So I'm going to tell you a story that is focused now about the host, but we're actually applying exactly the same method to the virus itself. The host story just matured faster, so this is what I'm going to share, but the method is the same. Okay, so what really transformed our ability to define genes in the nuclear membrane. And CMV, like all other herpesviruses, have a unique process of nuclear egress out of the nucleus. So herpesviruses actually build their new capsids, which are huge, in the nucleus, and then these capsids need to egress out of the nucleus. And it's thought to be a viral-dependent mechanism, so there are few viral, highly conserved herpesvirus proteins that are important for this process. So since NDC1 is important for nuclear pores, and we saw that it has an effect on the secretion, we were wondering whether it's playing a role in this process, and indeed it is. So when you take control cells and infect them with a virus that has a capsid or GFP tag, and you can see that 72 hours post-infection, as expected, most of the viral particles are now in this assembly compartment in the cytosol. But if we knock NDC1, you can see that now a lot of the capsids are still captured in the nucleus. So this proves that NDC1 actually important for the egress of CMV out of the nucleus. You can also quantify the viral genome. So we are trying to understand what exactly it's doing. Okay, so I'll just summarize what I just showed you. So we think VICOS is a really sensitive method to screen genes involved in CMV infection post-entry. I have to say, nothing that we've done here is unique to CMV, and it could be applied to all other herpesviruses. Probably it would be easier because the genome is more amenable for cloning. We can pinpoint the specific functions during the life cycle that each population affect. And we think it's provide really exciting opportunities to conduct screening in many different cell types and infection models, because we actually infect in very low MOI. So we don't need all the cells to be infected. So we are now applying this method to other cell types besides fibroblasts. And as I told you, we are now using a really similar approach to probe the effect of viral genes on viral propagation. So guide RNA can actually, this guide RNA can also target the viral genome because it doesn't target DNA virus. And then in this systematic manner, we can define the function of different viral elements. So this is something we are very excited about and we are doing now. So just to thank the amazing people that carried this work. So the initial shot of work was done by Avi and Yara. And then later on, the NSP1 characterization was done by Tal and Avi, two graduate students, in collaboration with the group of Shinji Makino. And the hamsters were done, hamster work that they didn't show was done in the Shioro Kuaka lab in Wisconsin. And all the work with SARS-CoV-2 was done in close collaboration with the Israel Institute of Biological Research. And then the VICOS work was really a courageous project of Yara Finkel and amazing graduate students that just graduated. And of course, thanks for funding and for you for listening.
Hi, very exciting talk. We are following a similar strategy to actually study HIV infection, HIV latency by encoding guide RNAs in HIV genome. much simpler to clone, sure. I was wondering about two things. One is how, when you infect cells with your virus, it must be a lot of volume, right? How you, for doing next infection to make it efficient, how do you, the simplification concentrated, or just, put, there must be some loss in that process as well, that affecting, your distribution of guides. And the second question is that do you think that still entry, you're still measuring entry because you're doing multiple rounds of infection, and what comes out of cell is affecting two functions, entry and something else, then those things will get depleted faster.
So I'll start with the second question because it's easier. So I don't think we measure entry at all because we right now target the human genome, right? So a virus, it's a wild type virus, enters the cells, target the human gene. The virus is still wild type. Doing what it's doing, it's budding out, it's a wild type virus. Now it's come to a new cell. The new cell, the gene is wild type, right? The gene that was targeted, this virus doesn't remember which gene it was targeting. So we're completely missing it. We didn't get any of the known entry receptor. Theoretically, what we can get is processes that affect entry by modulating the viral proteins, right? So this is kind of like affecting entry, but we don't probe entry at all. So I think it's really complementary to the screens that were done before that covered mostly entry and early steps. We're completely blind to entry. So that's the first question. Regarding the second question. Regarding the first question, so there are two steps. The first step, we have the bags with the guide RNAs. We need to reconstitute them to get a viral stock that really nicely represent the library we have. It took us a long time to learn how to do it. But once we use this adenofaction that I was just talking about, we can show that the viruses we generate fully recapitulate the bags we have. So we don't lose any complexity of the library. And now when we do the screen, we do it like in 15 big flasks, every replicate. So I think our data really show it. I didn't show you the guides are under control, but they are like flat all along the screen. So we don't have bottlenecks. Of course, in every screen, the question is, do you have bottlenecks? And this will, reduce the significance of your finding. But if you look at closely, the cell screen, we started to see some, potentially some noise. But this is why you have significance. And so our library is not a genome-wide library. It was more from technical issues in the beginning that it was really hard for us to clone a full library. I'm not sure we want to do it. I think actually doing it on 2,000 genes, even if we can go for 5,000 genes, is enough. So it's always a compensation, right? Do you go genome-wide and then use resolution? We had a really, really nice coverage all along the screen.
Fantastic, Dr. Really enjoyed it. So regarding all these genes that infect infectiousness, would this be that these proteins are being incorporated into the virion?
So I think this is the most fascinating. I think some of them maybe. We are really interested in gene expression, and I was actually surprised that many of the genes that affect gene expression per se are affecting infectiousness. So I think and we did some aspect. It's not the capsid composition, right? It's not so I think it is some things that are coming with the virus, but we were trying to figure it out. I have to say, it's to me, it's like, so what do you call a particle, right? So I'm saying these are particles, but the only thing I can say is that these are things that the genome is protected, because we put DNA, so we know the genome is protected. It could be that we have a lot of immature particles that carry the genome. So it's more like, the percentage of, and actually when you look at it, afterwards I start reading again, you look at all literature, we know that only 1 every 10 or 20 particles is infectious. So it's clear that there is a lot of regulation on the infectivity of the particles. For me, I was completely blind to that. I didn't fully think about it, but retrospectively, maybe it's not that surprising that this is where a lot of the regulation is going on, and we're trying to answer this, doing proteomics, trying to do EM, and to understand better what does it actually mean changing the infections. I don't have good answers, just...
Do you think that's the reason why CMV has such a huge tegument with so many proteins in it?
Part of it. I don't think this is... So again, looking at the literature on herpesviruses, it's known that by the way, not just herpesviruses, most viruses, the amount of infectious particles versus the amount of the particles, which are There's big, so I don't think it's unique to CMV. It's still we are looking at this. It's still an open question.
Thank you.
I have two questions, one for CMV and one for SARS-CoV-2. For SARS-CoV-2, I have been following that SARS-CoV-2 prevents nuclear export of mRNA since you gave the talk at Systems Virology seminar.
It was Yara.
Oh, it wasn't, okay, but it was your work in 2021, she organized it. So do you think that could be one of the reasons why the DNA vaccines didn't work very efficiently for SARS-CoV-2, or could that be one of the reasons?
Excellent. Because I have to say, I was super skeptic that it's inhibit export. And I was wrong. It does inhibit export, and NSP1 inhibit export. It's hard for me to say. Maybe.
Yeah.
I mean, but I think DNA vaccine failed beyond SARS-CoV-2.
Beyond SARS-CoV-2. That's true. But that could be one of the reasons adding to for the SARS-CoV-2. And the second question is, for the CRISPR ice cream that you did for CMV was like a lot of work. Have you tested it for latency?
So,
Also
latency is what keeps me awake at night. Sadly.
Same.
So basically the guides RNA are not expressed in latency. So I think what we can ask in the NSND model is once we reactivate the virus, do we see differences? But over there we have major issues of bottlenecks because only few cells are reactivated. So I'm still, the nice thing about it, this is why we call it so we can use this, once we clone the guide RNAs, which is a lot of work, we can use it now for activating screens, inhibitory screens. So I think maybe activating screens could potentially maybe start addressing things. I'm more, I think, on the virus side. So, if you activate different viral genes, how is this affect reactivation for latency? So this is the things we are now starting to explore. I wish we could use this system in latency, but I don't think so.
A great talk. I have a question regarding the NSP1 part. Did you have the chance to look at cellular interaction partners to understand the mechanisms as how NSP1 is managing all these different tasks?
So there are, I think, by now 10 different papers that including Nathan Corvin's, which was the first paper doing pull down on NSP1, also and looking at partners. You can find everything. The major question about NSP1, which again something we are actively working on, but there are also some publications that are now shaping what we talk, is that how is it degrading RNA? So for a long time, I don't know. What was in the literature is the idea was that it's somehow we put in cellular endonuclease to degrade cellular RNA because it doesn't have any nucleus domain. Some recent work, mostly biochemical in vitro, suggests that it maybe is acting as an endonuclease by itself. I'm not 100% sure what it means. What we are doing is we did a very thorough genomic screen to try and find things that support the RNA degradation activity of NSP1. We find something really interesting, but it's not the endonuclease. So maybe there is no endonuclease, maybe it is itself the nucleus. I'm not sure anymore. Did I answer your question?
Yeah, very interesting. Thank you.
That was a great talk. I really enjoyed it. I had a question about the first part of the talk. So I found it really interesting in your data that SARS-CoV-2 is shutting down translation, not primarily to promote its own protein production, but rather to avoid the interferon-stimulated gene response. And so, so many other viruses also shut down protein translation. Do you think it's the case for those viruses?
So actually, when you look at the literature, when people actually look at this, it seems like this is the major. So in HSV, there's all literature that actually show that if you infect interferon alpha receptor knockout mice, you can rescue there is a shutter factor of the you can rescue out of the effect. There are other anecdotal examples in the literature. So I think so. I think it's basically mainly blocking innate immunity, or at least this is the more critical step. I think maybe for SARS-CoV-2, because it's such a fast virus, it's even more important. But yes, going back into literature, all literature, I don't see any clear evidence that for the first one to support viral resistance. Right, thank you very much.
Oh, yeah, that's right. I was wondering with the, which of the first, yeah.
Finish off this presentation.
Oh, yeah.
Usually what we do in the lab is once we finish speaking.
Little meeting.